Hello friends, I am Akash Raut and welcome to Akash Biology. In today's lectures, we are dealing about one of the most important topic of botany, that is what mycoplasma. So taking no more time going on to the topic, first of all we have to know what actually is mycoplasma and what is the history behind the discovery of this unique organism. So in 18th century, in Europe there was a disease known as bovine pneumonia was taking place in the cattle, and that has created a havoc in all over the Europe, because beef and buff are the most important meat products that are being sold in the Europe. That's why, first of all, going on to the history, what we can see, so there was bovine pneumonia in cattle. So there was a disease known as bovine pneumonia. It is a respiratory disease, pneumonia, in cattle in 18th century in the case of Europe, and that has created havoc in all over the Europe. And later on it was found, initially it was thought that this disease is caused by a virus, but later on, after various experimental verifications and the discovery of various scientists, it has been proven that it is caused by organism that is neither bacteria nor it is a virus, and that unique organism is none other than what? Mycoplasma. So now coming on to some of the history factors. First of all, Louis Pasteur, that is the father of what? Microbiology. It occurs in reference to the respiratory system. So the Pasteur obtained the pleural fluid. Pleural fluid. Pleural fluid is the fluid that is found in between the visceral and the parietal pleura of the lungs. So if this is lungs, so this is visceral pleura and this is parietal pleura. In between there is pleural fluid. That sole function is to reduce the friction and to maintain the negative pressure inside the pleura. So he obtained the pleural fluid, but that pleural fluid was not obtained. So he discovered pleural fluid. He obtained the pleural fluid and named that organism what? Pneumonia like organism. That is what? PPLO. So the PPLO was named what? Pasteur, but he was unable to obtain this mycoplasma in pure form. That is, he was unable to obtain it in pure form. Later on, Nocard and Rocks obtained the mycoplasma in pure form. Mycoplasma in pure form? These are the mind-tickling questions that are being asked in the entrance exams. Later on, Borel named mycoplasma at what? Stereococcus. Stereococcus. And the name mycoplasma was given what? Novak. The name mycoplasma was given by Novak. So this is what the various terminology or the various discovery that was made by various scientists with respect to the mycoplasma. First of all, Pasteur, who named mycoplasma as PPLO. Second, Nocard and Rocks in the pure form. Borel named it as Stereococcus, and later on Novak coined the main term was mycoplasma. Another thing, if we have to give the definitions of mycoplasma, what actually is mycoplasma? So it is the smallest aerobic prokaryotic organism without cell wall. Without cell wall. So the main difference is what? It is the smallest and it is without cell wall. So it doesn't have the cell wall, and it doesn't have the cell wall means what? It is totally covered by cell membrane, so it doesn't have a proper shape, because the main function of cell wall is to provide rigid structure to any of the cell or plant cell. That's why they are pleomorphic. Pleomorphic means they are found in various forms, either coccus form or filamentous form, spirillum form or round form or oval. So there are lots of forms of the mycoplasma. So this is the pleomorphic. That's why it is often known as joker of plant kingdom. Joker of plant kingdom. joker of microbiology, sorry, joker of microbiology. It is not that the joker of the dark knight, it is joker of what? Microbiology. Another thing, there are lots of the features that the mycoplasma shares. Means first of all, it is without cell wall. Second one is they have colonial appearance, means they do not occur singly, they occur in what? Groups or bundles. So they have colonial appearance, and then after that they can what? As they are the smallest organism, they can what? Be filtered through the filter paper. So how much thickness it can? It can be what? It can be filtered through 450 nanometer membrane, bacterial filters. It can pass through bacterial filters. And the smallest mycoplasma is what? Mycoplasma has lots of species. Among them, there is one of the species which is very small, or the smallest is the Mycoplasma septicum. Mycoplasma gallisepticum. So it is the smallest non-mycoplasma. So this is about what? General features of the mycoplasma, and it has reported to cause various plant as well as animal diseases that I will be dealing later on. Now there is another thing, culture. What? Culture. What is the significance of culture? Means if we obtain any of the mycoplasma from the patient, then we have to what? Flourish it, or we have to see the colony for the various testing. That's why we do culture, means to multiply their numbers. So it can be multiplied both in living as well as non-living medium, but it can be flourished mainly in the non-living medium. So non-living medium, so what are the things that are to be the constituents of the non-living medium for the culture of mycoplasma? So it must contain what? Blood serum, therefore agar from red algae, and pH of what? 7.8. So we can see it can flourish in the pH of what? Basicity pH. If in the living medium, it grows sparsely in the living medium, and the thing that has to be supplied in the living medium is what? Chicken embryo. What? Chicken embryo. So these are the things for the cultures. Now coming on to the what? The structure of mycoplasma. What? A structure of mycoplasma. It is a very simple organism, and it doesn't contain what? The cell wall. So it is what? Cell membrane, and inside the cell membrane we can see a circular DNA. What? A circular DNA, and this DNA is double stranded. So it can be what? It varies from, as we have told earlier, it is varies from it is a pleomorphic, so it may be filamentous or it may be coccus form. So filamentous form is Mycoplasma mycoids. Mycoplasma mycoids is filamentous form. What? Filamentous form. Second one is coccus form is Mycoplasma pneumoniae. Mycoplasma pneumoniae is what? It is the coccus form, means coccus form. It is what? Filamentous form. As it doesn't contain cell wall, so it contains a cell membrane, and cell membrane is what? 3 layered, same as that of the universal layer, that is lipid protein lipid, and it is often known as what? Tripartite. Partitate means it contains what? 3 layers. So 3 layers of lipoproteins. And in the resting stage, sometimes what happens? So it is sometimes resting, means it is not being multiplying or invading anything. So the plasma membrane is enfolded to form what? Mesosomes. What? Enfolded to form mesosomes. But it is not present in the motile forms. Only in the stationary form the mesosomes are present. In respect of what we can see, the DNA is circular, long, double stranded. What? Double stranded long circular DNA. Then again there is what? Ribosome, that is 70s type, that is present in both the nucleus as well as in the case of what? Cytoplasm. And if we see, the DNA content is half of RNA. What? RNA. So the RNA is 8% and the DNA is what, 4% only in the case of Mycoplasma. The ribosome is what, 70s type, both cytoplasm is nuclear, and the DNA is what, long, circular, long and circular, and the main is what, double standard DNA, double standard DNA, and it contains various types of enzymes that are not so important. Therefore, another things, the thing that we have to differentiate between L-bacteria and the Mycoplasma. What L-bacteria and Mycoplasma? So first of all, what we have to know, what actually is L-bacteria. So during multiplications, when a bacteria undergoes multiplication for various generations, what happens, in some of the bacteria there occurs what, mutations in the DNA, and in L-bacteria there is what, no cell wall formation takes place, means there is no cell wall. Either the conditions may be unfavorable or there is a mutations in the DNA, any may be the reasons. So the L-bacteria and Mycoplasma both has absence of what, cell wall. But what is the main difference? How can we differentiate it is a L-bacteria or Mycoplasma? So what is the main difference is, if we place the L-bacteria in a sufficient medium, in favorable condition, then it will develop cell wall. What, it will develop cell wall. But the Mycoplasma will never develop cell wall. It is a myth that we have to remember. That is the main difference, and it is quite being tricky in the entrance examinations that we tend to forget. Another thing is what, the reproduction. So it occurs, the reproductions occurs by what, the binary fissions. Reproductions occurs by what, budding. It is simple, I will not be elaborating. Then another one is what, the fragmentation and binary fissions. What, binary fission. And in binary fission there will what, blebs and infrablebs formations. Blebs means, blebing means what, just a what, protrusion of the part of the cells, and bleb formations. And another thing that we have to know about the various diseases that are being caused by what, Mycoplasma. So the disease will affect both the man as well as what, the plants, man as well as the plants. So I have to see all the disease names. And the thing that we have to, on the basis of evolutionary sequence, first of all the virus has been evoluted, then after that Mycoplasma, then after that bacteria. So first of all virus, then Mycoplasma, then bacteria. Now coming on to the various diseases. In the humans, Mycoplasma hominis means mainly the what, invading the humans. It causes pleuropneumonia, then after inflammations in genitals, and similarly what, the infertility. Whereas Mycoplasma pneumonia causes atypical pneumonia. What, the primary atypical pneumonia. So these are what, the human diseases. Now in exams we are mainly being asked about what, the plant disease. So I will be listing all of these, and you need to mug up. So first of all is what, the London sewage. If you know any of the mnemonics, so you can post there, so maximum the students can get benefited from that. Disease of brinjal and tomato. So this one is the first. Second one is little leaf of brinjal. Third one is large bud in tomato, yellow aster, clover dwarf. Then after that what, bunchy top of papaya. There is one mulberry maize stunt. maize stunt, mulberry dwarf, and last one is what? Potato witch broom. Potato witch broom. As we all know, the penicillin mainly acts on the cell wall. What penicillin will act on the cell wall, so you can know about the trick. As it does not contain cell wall, so it will not be invaded by penicillin. So sometimes extra questions may be asked. So it is not been invaded by what? Penicillin. Not been invaded by penicillin. Why? Because the penicillin mainly acts on what? The cell wall synthesis, but it doesn't want cell wall. Then why it has to have fear with the penicillin? But the chloramphenicol, tetracycline and streptomycins all will act on what? The protein synthesis mechanism. So the mycoplasma can be invaded by, if you are going to be a doctor, you need to know if any person is being infected with mycoplasma, so you won't be prescribing the penicillin, because penicillin will have no effect on it and it is insensitive to that. But it can be invaded by what? The chloramphenicol, tetracycline, streptomycin. So this video is all about mycoplasma. At this short video, I want to share you one story that I have heard in what ISKCON that might be helpful to you. I will not take more time. I will take only 2 or 3 minutes. So once upon a time, on a sunny day, there was a farmer along with his 3 donkeys were heading toward the town, and the farmer has to sell 3 of the donkeys. But on the way, he saw a pond and wanted to have a dip on it. But to his bad luck, what happened? He has got only 2 ropes to tie the donkeys to the tree. As there are 3 donkeys, so he was confused and perplexed or flummoxed what to do now. He has got only 2 ropes. If we'll tie 2 and untie one, then one might go from there. So he was so much confused, and to his luck, one wise man was passing from his side. Then the wise man asked, "What happened to you?" Then he elaborated all his conditions. Then wise man told, "You don't have to be worried about that. What you have to do, if you have 2 ropes, first of all, what do? You will tie the 2 of the donkeys in the tree, making sure that the third donkey is watching that. And the third donkey, you have to what? Tie it virtually, means you have just to show that I am tying you." Then the man was what? Are you freaking crazy? The donkey will pass away if I do like that. Then the wise man told him that if the monkey will pass away, I will pay you. And then after doing that, he went on the pond and taking a dip, and after he came, to his amazement, what happened? The monkey was still there. So he untied 2 of the donkeys that he had tied and just started going towards the town. But the donkey which was virtually being tied was standstill. So he cajoled, he persuaded, he even bet up the donkey to move forward, but he was unable to move. He was again confused. What happened to the donkey? So he again called that wise man. What happened to him? What happened to that donkey? Then he told, "You haven't untied that donkey." Then again the farmer, to his amazement, replied, "Are you freaking crazy? I have not even tied. I have tied it virtually. Then why telling me to again untie that, even I have not tied in reality?" Then the wise man told him, "You know that the donkey has not been tied, but for the donkey it has been tied." So again he virtually untied, and then what happened? The donkey started running towards the town. So from this story, what we can take a moral, that in our life, what is that virtual barrier that is stopping us to move forward? That is what? Lack of resources, or lack of opportunities, or lack of what? Or there's a fear, or there's what? Hesitations, or there is what? A confusing, or there was a lot of expectations. So what are those virtual barriers which are just stopping us from moving forward? Or is there the what? Mental block? So in order to move forward, you have to untie that virtual barrier that is stopping you from moving forward. If we humans overcome that virtual barrier, we can accomplish anything. I can know about the situations of all the students preparing for the entrance examinations. They might be thinking all the students are scoring 90. What is my level over there? I will never get any seat if I do like that. And they started overthinking on the things that they don't have to think instead of studying. So my dear students or my brother or sisters, do not tie yourself in that virtual barrier as a donkey. Be the wise man, be the man to be reckoned with, because the thing that is going to happen depends upon the present hard work of yours. Stay tuned and connected with Akash Biology. And if this video is helpful to you, then please like, and for more videos tap the subscribe button. Thank you.